A short back info about Eneza. He's a fellow of the ENBIT, Masters at One University. He pursued his undergraduate in molecular biology and biotechnology from the University of Dar es Salaam, Tanzania. He has previously worked on understanding the population diversity of the whiteflies. Whitefly is host of cassava diseases causing viruses at Ayata, Tanzania, and is currently working on establishing and understanding the genetic basis of finger millet blast tolerance. His research interests are genomics, genetics, bioinformatics, phylogenetics, agriculture, and pathogen-host interaction. So today, Eneza will be taking us through his work, and we are looking forward to hearing what he has been doing. Thank you, Eneza. The floor is yours.
Okay, thanks for the introduction. Can you hear me?
Yeah, sure.
Okay, so I will share my screen. Yes, so yeah, so can you all see my screen?
Yes.
Yeah, so I am Eneza Mjema, and today I'll be taking you through my my MSc project, the project that I'm involved in, and it is titled Genome-wide Association Study to establish the genetic basis for blast infection tolerance and finger millet in Eastern Africa. Yeah, so I will start by introducing what is the finger millet and the aim and why we chose the finger millet. And basically, finger millet is an inbreeding allotetraploid crop with a genome size of about 1.4 gigabases. It is important staple food and source of income to millions of small farmers in Eastern, Southern, and Central Africa. And it grows well under minimal inputs on marginal lands, and its seed can be stored for up to 10 years. For that, it is a reliable food source, especially in terms of drought and crop failure. And it is high in calcium and zinc compared to other cereals. And thus, it is highly valued by breastfeeding women and usually used as a weaning food. Yeah, but finger millet produces a third of its genetic potential of 6 tons per hectare under irrigation. And most of the losses are accounted for by the finger millet blast infection, more than up to 80% in some of the pandemic areas in Eastern Africa. And to date, there are no reports on resistant varieties or QTL, that is quantitative tetrodotoxide, that are associated with the finger millet blast tolerance that have been seen to some of the farmers' preferred varieties. And so this project basically will be as a stepping stone towards the isolation of finger millet blast resistance gene. And that is basically for cloning and assisting the breeding for finger millet blast tolerance. So basically, for what this project, the analysis of this project is on the GWAS. And so GWAS is one of the approaches for marker trait association analysis. That is when you want to link a certain phenotype, say tolerance to blast fungus, and the genotype of the finger millet. Yeah, so you use GWAS as one of the tools that can show us if there is association to some of the SNP markers with our phenotype of interest. Yeah, and basically it uses SNP across the entire genome and the measurement of the phenotype. Yeah, so basically you need two kind of data, two type of data. That is the SNP data as the genotype, as you can see in the Venn diagram on the right side. So you need the genotype data. The genotype data for GWAS is basically the SNP data, and the phenotype data. These phenotype are the measurements, yeah, and using a model that is a mathematical expression, we can find if there is association of these genotypes to our phenotype of interest. Yeah? So, and in our analysis, each SNP are treated independently. Yeah. So this raises an issue of multiple testing. Yeah. From the basic statistics, we know that as the number of tests increases, the probability of getting a significant result by chance also increases. Yeah. So you can imagine in GWAS analysis, we have a lot of SNPs. Yeah. Let's say 10,000 SNPs. Yeah. And each SNP is treated as independent. So we have we normally have like 10,000 tests. So probability of getting significant SNPs just by chance is higher. So usually we do correction for multiple testing in GWAS analysis, and that some of the methods are used, either like Bonferroni correction or false discovery. Yeah. And so for the GWAS analysis, the two important outputs are the Manhattan and the QQ plot. Yeah, so we will see in subsequent slides the sum of these outputs from my analysis, and we will try to discuss them. Yeah, so this cartoon image here is showing them different models, like the general linear model, the mixed linear model, and the farm CPU. Yeah. And for my analysis, I've tested different models, but I've chosen to use the farm CPU. So the studies incorporate 4 countries in Eastern Africa, that is Ethiopia, Kenya, Uganda, and Tanzania. And basically for the genotype data, we had 167 diverse finger millet panel, which was previously collected under the BioInnovate project. And these accessions were genotyped using the genotype by sequence approach at the University of Georgia. And so SNPs were called and quite filtered was performed using the tassel unique pipeline. So we had around 10,000 SNPs, yeah, for the analysis. And for the phenotype data, blast scores phenotype data was obtained from an infection assay. And basically the infection assay, it just involved the inoculation of the fungus on the surface of the leaves. And for that, we had chosen 4 fungal isolates, each from the representative country, that is E43 from Ethiopia, K1 from Kenya, T1 from Tanzania, U8 from Uganda. And this was done at the Salisbury Laboratory in UK, and the visual blast scores were recorded. So as you can see on this image here, so we have scores, and the scores basically represent the level of the blast tolerance. So for instance, for level 1 is a resistant, as you can see. And for instance, the level 5 is highly susceptible. Yeah. So it's actually the damage, how the finger millet are damaged by the fungus. And for each isolate, we had 3 biological replicates. Yeah. So my approach was to start with a QC of the data, and that is basically trying to format my data in a format that will be used in the subsequent analysis. And since I received the data as in Excel format, so I had to convert them. And for instance, for the genotype data, it was encoded in the ABH format. So the first task was to actually to convert it in a ATCG, the normal code for DNA, and that been using Python together with R for that. And so the file format, that is the HapMap format for the genotype, and the text format for the measurements, that is the phenotype. Were used as the input for the quality control, and for that I use the tassel. And so prior to GWAS, that is association test, I had to do the population structure and kinship analysis. Yeah. So the population structure, that is PCs, together with the data that the genotype and phenotype were used as input for association test using the GAPI data package. So that's like the overview of what I've been doing for my analysis. Yeah, so and basically, so I will be taking you through some of the output of the analysis. And I'll be starting with the response to blast infection. And basically here, we were looking on the within these different levels of the visual blast scores, that is 1 to 5. And these are for fungal isolate that were used for analysis, like how many counts are within each bin, that is in level 1, 2, 3, 4, and 5. So for instance, for the E43 here, you can see that most of the counts are within 1 to 4. And basically we expected this because 1 and 5 are the outliers, and we know what we want is 1. And so basically we want to push the population mean towards the desired trait, which is tolerance. So tolerance for our case is like outlier. For an accession to be a tolerance is an outlier. So we expected this for the most of the these fungal isolate, the distribution, like the counts in each bin were quite well. But if you have notes, we like to take your attention to this T5 here. So this was basically, I can say, like a control, because this is a mutant strain of the fungus that affect rice. And these fungus have very high degree of specificity. And thus, if you have noticed, for the T5, most of the accessions were within the 1 to 2. So like they were high resistance to this isolate, and this was expected because this is with the fungal strain that infect the rice. Yeah, so basically, and for this, so I had to use tidyverse for data wrangling and plotting. Yeah, just to get the descriptive statistics. So these are bar plots, but I had other output for like Instagram and yeah, density plots. Yeah, so these are the linkage disequilibrium. So what is linkage disequilibrium? Linkage disequilibrium is just the non-random association of the markers, and that is like we know that some of the markers tend to be inherited together. Yeah, it's kind of the same as the concept of linkage, chromosomal linkage, that some of the genes tend to be inherited together. Yeah, so some of the markers tend to be inherited together most of the time. Yeah, and due to the presence of linkage in a genome, linkage disequilibrium of the markers, this help us to do GWAS. Because remember, I said that the genome of the finger millet is 1.5 gigabase. So it's not possible to genotype like all the SNPs that are within that genome. Yeah, so the presence of linkage disequilibrium help us to just to use single SNP within a certain block that are in high linkage disequilibrium as a representative of that block. Yeah. So basically, two ways of visualizing it: the LD decay on the left side, and the heat map on the right side. Yeah. And so for the finger millet, the LD extent is up to 300 kb, as you can see in the LD decay. So that in simple term, it means that if a SNP is in position 1, it is in LD to a SNP that in position 300 kb. Yeah, and for that reason, we can just, if our region is 1 to 300 kb, we can have 1 or 2 SNPs representative of that region. Yeah, because yeah, so in that that being said, it means that the SNPs that we are having for analysis were sufficient for GWAS analysis. Yeah, and so the heat map here, I've just taken a small portion of the chromosome 5 just to get the what it means. Like you can see here, this is the block 1, and the LD extend within this block up to 443 kb. Yeah, so you can see, so this LD show on average within the finger millet genome what is the extent of LD. And for, yeah, so this is just to answer that my SNPs that I'm having for my GWAS analysis, are they sufficient? Are they sufficient for a significance analysis? And basically, the answer is yeah, they were sufficient for GWAS analysis. And for this, I used Prink. Some of the analysis for the LD, I used GAPI, and for the to draw this heat map, I used the Haploview. So prior to GWAS, I had to conduct a population structure analysis. And this is basically the model that I'm using. It takes into account the population structure and the kinship within the markers. So, and for the population structure, I used the ADGenet R package, and Factor Extra was used for plotting these PCA. So basically, what this PCA are depicting is we had two major clusters. For instance, we are looking at this PCA1 versus PCA2. We had two major clusters, and yeah, so the one of the cluster on the right side is small. That's a small population compared to this one, which is larger, and it shows a continuous genetic diversity. And so my assumption so far is we know that finger millet was originated in Ethiopia and Uganda 5,000 years ago. So it could be possible that these small population here, which is quite different from this one, which is bigger, could be the wild types. Yeah. So, but yeah, but we are still doing some more analysis on looking on their contribution of these individuals markers, so as to have a concrete understanding of what causes these two distinct population, with one being very few compared to another, because could be their outliers. But yeah, so far my assumption is these are the wild types, and these ones are the ones that were domesticated. So over time, they have evolved and changed. So they clustered separately from this small population. So, and that was true for the plot for PCA1, PCA2 against PCA1, and PCA3 against PCA1. And if we are looking on the contribution of each principal component, that is PCAs, so 1 up to 10, so PCA1, 2, and 3, together explain the cumulative variance of 40%, and they were larger. So for my GWAS analysis, basically I had to use only the 3 PCAs as they represent the larger variation across the population. So as I said earlier, the two major like output from the GWAS, like the visualization, are the Manhattan plot and the QQ plot. And so also I use the GAPID R package for the GWAS analysis, and these outputs are from that R package. And so what we are seeing here, these are Manhattan plot associating the disease resistance phenotype across all the accession that were 167 finger millets. And each identified SNPs in Manhattan plot is represented as a dot. Yeah, so, and the x-axis is chromosome numbers. So for instance, in finger millet, we have 9 chromosomes, but it had chromosome A and B. So what we are seeing here, like A1, AB to A2B to A3B, these are chromosomes. And the y-axis is a negative log of p-values. Yeah, as I said earlier, also the p-value for a GWAS analysis is usually very small, very small. We can take an example. If we have if we have 1 million SNPs, if we have 1 million SNPs in a GWAS analysis, so the p-value after bond-for-RNA, for instance, bond-for-RNA correction, it will be 5 times 10 raised to -8. So we can't really report those very, very small number. That's why we use a negative log. So that's a transformation. So for my analysis, since I had almost 10,000 SNPs, but basically after filtering for some of SNPs that we are not reaching the threshold, I had almost the 9982, so 9,992 SNPs. And basically, so the threshold that is dividing the p-value 0.05 to this number of tests, which are the markers, you get around 5.4, like 5.4. So that is the green line, what you are seeing here. And this is the cutoff, yeah? So the SNPs that are above this line, these are significant SNPs. So we can say that these are SNPs or markers that are associated with our phenotype. Yeah, so for instance, for my analysis on finger millet, we had total nine markers that are associated with the tolerance, and 7 of the markers were present in chromosome 5. That is, for the U8, we had 2 markers at chromosome 5. For E43, sorry, and for U8, we had 2 markers also, this green here and this purple. And then for K1, we had around three markers at that chromosome 5A and 5B. Also, we had chromosome 1 marker in chromosome 2A of U8 and 1 marker in chromosome 1A of K1. Yeah, so basically that is what these plots are depicting. And this concur or aligns with our assumption that since the blast tolerance is a quantitative trait and the biology behind it is that the quantitative traits are controlled by QTLs, yeah? So these are genes in different locus, and all of them add like in additive fashion to the phenotype, which is a blast tolerance. So also for the QQ plot, and basically for the QQ plot, the x-axis is expected or theoretical p-values. The y-axis are the observed p-values. That these are the ones that we have visualized from the Manhattan, which is the model calculated for us. So basically here we can see if the model was was powerful in modeling the association in our data. Also, we can tell on the population structure. So basically what we are seeing here, for instance, this is for QQ for the E43, U8, T1, and K1. So most of the time we expect most of the data points at the beginning of the line, this regression best fit line, the beginning of the line to be within the most of the point to be within of the line, and they for the rows which have showed the significance tend to move away of the line, since the theoretical values are drawn, are calculated from a completely normal distribution, and the null hypothesis for the null hypothesis is true, and the null hypothesis for the GWAS being there is no association between markers which are SNPs to your phenotype. Yeah. So as you're going, at this point are going outside, these are the ones that didn't obey the assumption, and these are the ones that we are showing the significance to our phenotype, association to our phenotype. Yeah, so so I'd like to acknowledge my colleagues at Fabric, funders, and BHKi, my co-investors, and the entire team. Thank you.
Thank you, Eneza, for the nice presentation. I enjoyed the presentation, so now I open the floor for questions. So If you have a question, kindly unmute and ask the question. Okay, in the meantime, I'll go with the first question. So I was wondering, what criteria did you use to remove SNPs which are in ILD? In ILD, what criteria did you use to remove the SNPs?
So currently I haven't removed the SNP that in high LD because that is after I've gotten these significant SNPs, that the SNPs that are associated with the trait. So the next step will be to look which of these significant SNPs are in high LD with others. So that will guide me to remove the one that are in high LD. Yeah, but so I would have removed the SNP that are in high LD if I was doing let's say structure analysis. Yeah, so for structure analysis, basically you don't want SNPs that are very like in high LD, yeah, because they are basically telling the same story. So, but for the GWAS, at first you will have to use all the SNPs you are having, but the LD now for the GWAS, it will tell you if the SNPs you are having are significant for that analysis, depending on the genome size of your individual. Let's say for instance, for my analysis, yeah, as I've said earlier, the finger millet is 1.5 gigabases. 1.5 gigabases. So the SNP that I'm having initially were 10,000. Yeah. So the question someone will ask you like, okay, are these 10,000 SNPs like representative of these 1.5 gigabases? Yeah. So now for the GWAS LD will tell you this. Yeah. So for instance, if I'm saying like, okay, the extent is from 1 to 300 KB. Yeah. So ideally, I need one SNP in this window of 300 KB. So if I need one SNP in the window of 300 KB, how many SNPs I need in 1.5 gigabases? Yeah, so that comes around like 6,000. Yeah, so yeah, but yeah, going further, I will have to remove the SNPs that are in high LD. Yeah, that's next step actually.
Eneza, just to build up the discussion about the size of the genome, you say the estimate is 1.5 gigabytes. Is it the estimated size of the genome, or I mean, I'm just curious to know what is the current status of the assemblies of finger millet. Yeah, so that is one, and even to also know the cultivar that was used to come up with the the genotype data that you're using in your analysis. Was it from one of the East African countries? Yeah. And then another question is, you on your Manhattan plots, you mentioned, for example, the CPU U8, that there are two SNP markers that actually are associated to the resistance. Yeah, but I saw that there's also one that was above in chromosome 2B, and you mentioned only two. So why did you leave that out?
Okay. So yeah, so thanks for the questions. Yeah, so basically the size of the genome of the finger millet, because basically finger millet is one of the orphan crops. Yeah, so there are no like published the reference genome for the finger millet, but most of the papers that have been published, they use the reference that they used, which is not yet published on the databases. It's ranged from 1.4 to 1.5 gigabases. Yeah, so this is like estimated genome size. Yeah, so yeah, it's true. This is estimated genome size. So the assembly are still going on. So far I haven't yet met the paper that talks on the published the reference genome for the finger millet. And for the cultivars, so the sample that are that the finger millet samples, these one were retrieved from the BioInnovate project, yeah. And basically from the BioInnovate project, they were collecting all the finger millet cultivar in these 4 countries, yeah. And this project was around 2010, yeah. So they were collecting all the varieties, basically those mostly the preferred farmers, preferred variety by farmers. So they were collecting it in different those 4 countries, yeah. So yeah, so it's like it's a diverse panel, yeah. We have those from those 4 countries, but also we have other cultivars from India, Nepal, yeah, and other countries, yeah. So it's quite a big, but I haven't had many like information on the BioInnovate project. But yeah, so what I know that they collect the cultivars from all the 4 countries, and these cultivars were preferred by farmers. Yeah, so I can add more on that. Also, for the case of the Manhattan, I think I mentioned it, like I mentioned it, that one SNP in chromosome 2. I mentioned it. So if yeah, it's not that I've left it. Yeah, this one too. Yeah, I mentioned it, that one is SNP in chromosome 2 and one is SNP in chromosome 1 of the K1. Yeah.
Okay, thanks very much.
Okay.
And there's also a question on the chat.
Also,
Mike, can you
yeah,
go ahead?
so
Yeah. So the question on the chat, they're asking, what is your take on the variation within the PC1? Do you think the Ethiopian blast has been conserved over time based on the independent clustering?
so basically, Mike, that's one of my assumption, yeah, because we know that for sure finger millet, and most of the paper have been published in saying that finger millet was first originated in the highlands of Ethiopia and the western of Uganda. Yeah. So like the wild types of finger millet, probably they are in Ethiopia, Uganda, and probably in Kenya. Yeah. So that's basically my assumption. My assumption is this could be the wild types that have been conserved over time. And the other group, the bigger one, these are the one that after domestications and have undergone the selection pressure, like the breeding. And yeah, so it could be possible that these are the wild types that have been conserved over time, but also it could be possible these are outliers. So I'm still working around on finding a way to explain this, and they are looking like the news and others. So just to see maybe the contribution of each marker and to, yeah, so like, but basically that is my assumption. These are the conserved over time, and that's why they cluster themselves of that bigger population.
Thank you, Eneza.
It answers Mike's question. Or?
Maybe Mike can type in the chat to let us know if his question is answered. So in the meantime, I also have another question. So I was wondering, the farm CPU model, does it use all the SNPs or just uses a select number of SNPs? Or exactly, I know for the linear mixed models, there's a way you can fine tune your models to use either all the SNPs or just the SNPs which have the largest QTL. So how about the farm CPU model?
Yeah, so basically, for instance, for the mixed linear model, as you have said, you can fine tune and put it in a way that it can use maybe a representative of the SNPs. Yeah? But for instance, the limitation for the mixed linear model when it comes for the Q matrix and the K matrix, they remain the same, constant. Like the Q matrix with the population structure and the kinship matrix, they remain constant. And that is what the farm CPU tries to solve, yeah, because you can't keep these two matrices constant over entire of the analysis, because some of the significant SNPs you need to modify if you have gotten significant SNPs, yeah. So for the farm CPU, what happens for the farm CPU, the Q and the K, which so Q is for the fixed model, and K is for the random model within the linear model. These are two run separately but iteratively, yeah. So basically these tend to change as you get the significant SNP. So what that's basically what it does. And this is okay, I was on my opinion, this is good when you are actually working with an inbreeding organism, like for instance these crops, yeah, because the inbreeding tend to affect the way these maybe allele frequency and those how genes are being inherited. So for my analysis, the farm CPU uses all the SNPs but for the Q, for the calculation of the kinship matrices, it doesn't use all the SNPs, yeah? So it uses only the what we call quantitative trait nucleotide. So the only nucleotide that have shown significance. So it kind of modifying the kinship as the analysis going on. So that's why it separated the fixed and the random separately, but iteratively, yeah? So yeah, so basically it uses all the SNPs in other analysis, but it uses fewer SNPs for the calculation of the kinship matrices.
Thank you. I think that was quite clear. And that leads me to another question. What is the genetic architecture of the finger? Is it polygenic or sparse, the genetic architecture?
So basically, what I know on the finger millet, it is a polygenic, and it is a allotetraploid. Allotetraploid in a sense that it has a two, like, so basically finger millet is a result of breeding two distinct crops, yeah. So one crop with the chromosome A and another with chromosome B, but it is a tetraploid in a sense that A is duplicated, so it is AA, and B it is duplicated, so it has 4 sets of chromosomes, yeah. But yeah, so that's basically the genetic architecture of the finger millet, but it's the polygenic.
Okay, thank you. So any other questions from the members?
Can I go?
Yeah, sure.
Eneza,
Yes.
sorry, perhaps you didn't understand me on the questions that I asked. So if you go back to the PCA plot, please.
Okay.
Yeah. So if for the first figure, I know you're comparing PC1 and PC2 here. Yeah. So I think I missed it. I should have indicated PC2, not PC1. So if you just look at PC1 on the first figure, there are things that are far away at the top, and then the others that are far away at the bottom. What do you make of this large group that you have things that are sparse that much? You see, like for example, whatever is at the bottom is mostly from Ethiopia, and then whatever is at the top, you have groupings of what's from the rest of the region. What do you make of that? Thank you.
So basically, so my interpretation of this is that, so basically this is a large, this is a large group showing a continuous pattern of genetic diversity. Yeah. So, and if we try to Ethiopian population that they've from and other so this concur with the assumption that we had before, yeah, because we know that the Ethiopian actually, before the introduction of these finger millets to other part of the Eastern Africa and the world, they were originated from Ethiopia and Uganda, yeah. And I think we don't see most of the Ugandan sample because basically within the 167 sample, we had fewer Ugandan sample, yeah. But the Ethiopian grouped themselves together. Yeah, I agree with you. They have conserved over the time, and it's concur with our assumption that the finger millet were originated from Ethiopia, and that's why even in this small population here on the right side, there are samples from Uganda and Ethiopia. Yeah, there are samples from Uganda and Ethiopia. Yeah, so I agree with you that the Ethiopian samples have been conserved over time, and before doing it, we expected that there will be a clustering pattern of the Ethiopian and Ugandan samples, and the Tanzanian, Kenya, and non-Eastern Africa to be together, yeah, because before the introduction of fingerprint to those other places, it was originated from these two countries. And so, sorry, because we had fewer Ugandan samples, but in this down part of the axis, we expected to get the Ethiopian and the Ugandan samples.
All right, thank you.
Could I ask a question, or do we raise hands, or?
Yeah, sure. You can go ahead, Jennifer.
Could you bring up that last image?
Last?
Yeah, the last PCA plot that we were just looking at. I just wanted to make sure I understood. So it's really the PC1 that's separating the Ethiopian and Ugandan, right? Because it looks like it's separated by PC2, but really there's Ethiopian underneath the Tanzanian samples, right? Like the plotting kind of stacks over it. You see what I mean? It looks to me like there's like the dark green underneath, and that it's sort of an illusion of the plot that there's only Tanzanian above the axis.
So sorry, but basically this is because the sample size we are having, the sample size we have a lot of Ethiopian and Tanzanian samples compared to Kenyan, Ugandan, and the non-Eastern African samples. And it's true that the PCA1 was the one that we are able to cluster these two groups separately. But looking for the PCA2 and 3, it's kind of continuous diversity of these sessions.
This is really interesting. Is there like a publication that you can point to, or a pre-publication, or like a website for the research that you're doing that I could read more about it?
So currently there are no reports on finger millet diversity. So actually this is the first study. Yeah, so this is the first study on the GWAS for the finger millet and the population diversity using the SNPs, using the SNPs markers. This will be the first population diversity of the Eastern African finger millet.
Well, good luck. I look forward to reading more.
Yeah.
I think maybe Eneza, when your work will be out, maybe you can share so that people can get to read it. So Jennifer, I think at this point you'll have to be a little bit patient, because I think he's still working on it.
Definitely. I did some research on plant genomics earlier, and I always think it's
Yeah.
fascinating, you know, because like the way you were describing the different ploidies and the mixtures of different populations, it's very complex compared to like human genomics. But it's really fascinating.
Yeah. Yeah, so it's basically even I myself I'm facing the same challenges, yeah, like to get the, for instance, some of the paper that we can refer to talking about the finger millet in the Eastern Africa, yeah. So the challenge is always to make sense out of what I'm explaining, like, okay, does it maybe concur with the reality on the ground and stuff. So, but yeah, when we are ready and it's out there, we'll be sharing the outputs.
Festus, maybe this can sound a naive question. I have not drawn these PC plots before, and interesting to see a good clustering and all that. So, what is really good practice when you're comparing PSCA 1 and 2, 1 and 3? To what extent? Because I think this can even go away to PC10. I don't know, as in, what would you advise someone who's doing population structure?
Eneza.
So yeah, so yeah, so it's true. Yeah, it's true. Usually, usually when you like maybe okay, maybe if you are using a package, maybe say the base R, the PR comp, usually it will calculate for you. Okay, if you haven't maybe put the default, okay, you will say maybe I want 10 first PC. Yeah. So what dictates you to use maybe PCA 1, 2, and 3 is this what we call script plot, yeah?
Okay.
So the script plot show the percentage of the explained variance. Like, okay, I have PCA 1 up to PCA 10, yeah? But PCA 1, the contribution, like the variance explained by PCA 1 itself is way bigger than adding up to PCA 5 up to 10, yeah? So basically, yeah, so basically I can just use PCA 1, PCA 2, and PCA 3, and then I will get 40% of the explained, actually 50% of the explained variance, rather than adding up all the other PC, yeah? So
okay,
basically, yeah, so it calculates for you many PC, like, okay, up to 10, depending on the, okay, depending on the number of PC you want to analysis. So if it's 5, it will calculate PC 1 up to 5. But again, what you have to do is to check on the percentage of the explained variance for each PC, yeah? And from there that, okay, for instance, if I had quite a small PC explained by these 3, I could have gone up to 5, yeah? That's why in a GWAS, it's like in a GWAS analysis, you can say that, okay, now I want to use only the 3 PC, or okay, I want to use only the 5 PC, because okay, adding up these 5 PC, it explains the much of the variation, yeah? So that's what dictates you on the saying, okay, I want to use PCA 1 and 2, I want to use PCA 1, 2, and 3, yeah.
Okay, interesting, thanks.
Yes, yeah. But also you can go further. You can go further and check if, okay, let's say, okay, I have markers. For instance, I have 10,000 markers, yeah? But within these markers, which markers are they, like, have the highest contribution in the variation, yeah? Okay, you may have a lot of markers, but those markers didn't tell anything about the variation, yeah? So you can even go further and exclude markers that doesn't have like a high impact on the variation that is being observed, yeah? Also, you can go further and check maybe some of the contribution are very high, so they could be outliers and stuff. So yeah, it's quite it's up on you on understanding what you want to do. But yeah, it's quite free. You can do anything you want. But yeah, that will be my answer.
Thank you, Eneza. Just a quick one. What tool did you use to calculate the PCs from the genetics, from the populations?
Yeah, so for the PCA, I used the Adigenet error package. So basically, you have, for instance, for my analysis, I had the HapMap file. Okay, so I had the Excel. I had to find a way to convert it into a HapMap file. So this HapMap file have the ATCG. Yeah? but for the input data for the adigenate, for instance, is a binary file. Yeah? So I had to convert them into zeros and ones, and using the adigenate error package, it calculates for you the PC. So you use the factor extra error package for plotting those PC output. But I could have also used the PR comp, for instance, the base error utility to calculate the PC. But yeah, basically for me, for my analysis, I use the adigenate error package.
Awesome, thank you. Any other burning question or suggestions or comments? Okay. I think that was a nice presentation, Eneza. So at this point, I'd like to let people show you some love by putting on their videos. I think for one photo for your archives and for BHKi. Kindly, you can put on your video.